What's it like working in an agricultural research lab? Is it really a good opportunity? We might be a little biased, but working for the USDA can be a rewarding and engaging opportunity, whether you're looking at long or short-term career options, or even if just you're looking at an internship to get more experience or decide if this career path is right for you. Each lab is different and has its own unique culture, but in general, project successes and failures are a team effort, because in the end, science is all about collaborating and connecting. Hi, my name is Rachel Nagley, and I'm a research horticulturalist with the USDA ARS in Parlier, California. So my lab specializes in grapes, specifically abiotic and biotic stress resistance. And what that means is we basically study how grapes respond to the changing environment. But almost everything can be affected or is affected by abiotic and biotic stressors. So this can be things like insects, diseases, heat, drought, even fruit quality could be affected by stressors. So if you have a high quality grape that is going to produce nice, beautiful clusters, if you give it too much heat, you don't give enough water, you expose it to disease or insects, your fruit quality is going to go down. And sometimes when you're breeding new varieties, you incorporate disease resistance from wild species. And a lot of times these wild species have fruit traits you don't really want. They might have seeds, or they might be very bitter, or have really thick skins, things that you don't want in something like a table grape. And because of this, my lab touches on a lot of these different areas because they're all related. So today's presentation is going to be more of an overview of the different types of research projects that my lab does. But I also want to introduce you to some of the different people who participate in my lab, because science and working in a lab is so much more than just a single person working in a vacuum. So in addition to hearing about my own path to science, I'm also going to introduce you to other members of the lab. These are people who are students, they're technical staff, they're early career researchers, and they'll tell you a little bit about the projects they work on, their career path into science, some obstacles that they had to overcome, some advice, and hopefully they'll give you a different perspective on what it's like to be in science, depending on what your education and career level is. So we're based at the San Joaquin Valley Agricultural Sciences Center in Parlier, California. There are about 20 different PIs at this location working on a number of different research topics. For example, there's a lab that's focused on breeding new varieties of raisins, table grapes, almonds, and apricots. And in their work, they work to develop raisins with fun flavors and softer wrinkles, almonds that have higher yields, and apricots and plums with bigger fruit, better flavor, and always disease resistance. There are water management labs that work with tomatoes, grapes, pomegranates, and they cover everything from water use efficiency to soil microbial community composition and how water can affect that. There's a unit dedicated to post-harvest diseases and insects. There's even a germplasm repository where genetic material is collected around the world for waiuli, jojoba, puntia, and a few other species. And these are maintained and made available to other scientists around the world. These projects range from highly molecular or sequencing-based to highly applied, such as determining insecticide spray recommendations. But this is just the diversity of labs and research that can be found at just one ARS location. So my lab is what you would call a translational lab. And what that means is that instead of doing basic research or doing applied research, we do something in the middle and take a little bit of both. So with basic research, you're generally trying to answer a very scientific question, whereas in applied research, you're often trying to solve a very real-world problem directly. It's not that basic science doesn't contribute to solving real-world problems, it doesn't tend to have as an immediate application. Translational research tries to marry those two types of research. So in our case, we do a little bit of field work, we do a little bit of lab work, and most of our projects are some kind of combination of those two.
My name is Aaron Rocha. I'm an undergraduate at Fresno State, majoring in plant science, crop production. Here in Dr. Negley's lab, I am a field assistant. Working here in Dr. Negley's lab, I'm part of the maintenance crew. What my job consists of is maintaining vines in greenhouse and in fields that are used for research projects. To transplant the green cuttings that we pulled out of the rooting medium, we first fill up these cylindrical pots with potting soil. Then we carefully plant the cuttings without breaking the delicate roots. These will then be used for the mealybug resistant test. I didn't go to science intentionally. During my first year at Hartnell College, I had planned on becoming a PCA. I like the idea of having my job consist of identifying pest issues and providing solutions to farmers. I had my mind set on that goal up until my second year at college. One day I walked into my plant science instructor's office with a question about an assignment. Before I could say anything, she told me she had sent my contact information to the plant pathologist in charge of the plant virology lab over at the Salinas USDA station down the road. I knew that they had an opening for an internship. I just didn't apply because I thought I wasn't qualified for it. I went through the interview process, and a week later I was hired, and I ended up working there for about 2 years. During those 2 years, I maintained live virus cultures and insect colonies. I also conducted several disease-resistant and host range experiments with various plant viruses. By the end of the internship, my passion and interest for plant diseases and insects had grown. Of course, as with everyone else, there were obstacles I had to overcome. The biggest one was me. I would think negatively about myself and I would doubt my abilities. Over time, I learned to have confidence in myself. I started to have confidence in my skills and abilities. I now know that even without the necessary experience, I'm still able to excel at a job if I believe in myself.
So I'm what you would call a PI, or a principal investigator. And basically that just means that I'm the lead scientist for the lab. I run the lab, I do a lot of the higher level activities like administrative work, I write grants, I help manage people, I help kind of direct where our projects are going, and I oversee a lot of the bigger picture things. I still do data collection, I still go into the lab if I needed to, or I still collect data sometimes, but a lot of times that's only if we're short-staffed. In general, most of the data collection and the day-to-day activities and the science is done by other people in the lab. And where you fit in the lab and the activities that you do, a lot of that is dependent on your education level.
Hi, my name is Susan. I am a biological science technician here at our research station in Parlier. I work with pathogenic fungi like powdery mildew on a variety of crop species like roses, grapes, and peaches. So a lot of what I do is collecting swabs of powdery mildew from the different species that we work with, or getting swabs in from our project partners, and then doing Chelax DNA extractions on these so that we can determine if there's differences in fungicide resistance between isolates or between fungicide treatments used. A really big reason for why I decided to pursue a career in science was my sense of responsibility for the land around me. I think that it's very important to protect and uphold what you care about, and I really care about natural spaces. I think that it's a really good way to make a difference. Being part of the science is one of the most important things that I feel that I can do to help preserve and protect these places for everyone's enjoyment in the future. I know that agricultural landscapes make up a lot of the natural world around us, so it's really important to make sure that these spaces minimize their impact on natural ecosystems. I grew up on a small farm, and it really helped me to develop a sense of stewardship at a young age. And I think that this stewardship really gave me a sense of responsibility. I think that being part of the science that surrounds farm management is really important. If we can help farmers to make better strategies for managing their land, then we can probably reduce their environmental impact.
Right now we're in the process of collecting isolates of powdery mildew from peaches, and to do this we need a lot of seedlings to transfer the fungus to. We decided to grow our own seedlings, so we went out into our orchard and collected hundreds of peaches for their seeds. A really fun part of this project was breaking open all of the peach fits to find seeds that looked just like almonds, which I definitely wasn't expecting. Not all of the seedlings survive, so you have to learn to identify the bad ones, which isn't that hard considering that they look like this.
I did face obstacles to get where I am today. I was a first-generation student, so I had to build my own resources to rely upon since my family didn't really know what I was going through. I also was a low-income student, so while I did qualify for FAFSA and that helped a great deal, it didn't quite cover the cost of living, and I had to have multiple jobs pretty much my entire time in college. This impacted my ability to participate in as many extracurriculars as I wanted, and it also impacted my ability as a student, and I really had to wedge my study time wherever I could get it, whenever that was. I would love to end up as a professor because I know no matter what age you enter undergrad, a lot can be uncertain and you might not know what you want to do. I think that teaching classes that I'm passionate about might help to spark passion in others, and it might make someone more sure of what their career goals are.
One of the projects that best exemplifies this idea of translational research is our Xylella project. So this is a project where we are evaluating more than 20 different strains of bacteria that cause Pierce disease on grape to see how much disease they cause and if there are any differences. For this project, we inoculated these plants and we're evaluating them for disease symptoms over time. The symptoms that we're typically evaluating are leaf symptoms. So you can have some red edges to your leaves, you could have necrotic lesions, or the leaf can fall off entirely and you're left with petioles. So these experiments can take place in the greenhouse or the screenhouse and typically take about 9 weeks to complete. So after we've done evaluations, we then have to process samples. And to process those samples, we end up taking each plant and collecting petioles off them. Now this petiole collection happens whether the plant is alive or dead because we're going to return these to the labs for DNA extraction and to actually evaluate and see if there are living bacteria in them. So Susan's kind of demonstrating what happens with the petiole collection here. We take each plant, you break off the leaf blade, and then you take the petiole and you put that petiole inside one of those envelopes. And those envelopes are going to go back to the lab for processing. Now, as I said, we do it for qPCR analysis, but we also do it for extracting bacteria. So if we were extracting bacteria, we take those petioles, we put them into these bags, these mesh bags, and then we grind them up using a pestle. Once we've done this, we then add a little bit of buffer as well to help create enough liquid, and then we pipette out liquid and we put it into those tubes. And then we're going to use those to put onto the plates. So this is just to show you what it looks like. In this case, we are taking one of our sets of dilutions and we are pipetting it onto a PD3 media plate. We're going to put 2 droplets on each dilution. We're going to let them gently slide down the plate, and then we'll come back a couple weeks later and we'll evaluate them to see if there was any bacteria that grew. So I said that translational work takes greenhouse, lab work, and matches it with field work. Well, the field is where things start to get a little bit more interesting and challenging.
Hi, I'm Alana. I am a biological science technician here at the ARS, and I was asked to talk a little bit about how I became interested in science. I'd say that I didn't start out knowing what I wanted to do. Basically, starting in my senior year, I got involved with a restoration project that had to do with an endangered species of vole. I know, a very strange animal to get involved with, and to make it stranger, it was a vole that lived in a wetland in the middle of the Mojave Desert. This project definitely is a place where my passion grew for restoration. I'm a vineyard and greenhouse technician here at the ARS, and as part of my duties, I'm in charge of propagation, maintenance, and prepping and running projects during the summer. The summer is our busiest time of year. This is when we get crosses from our previous year planted, when we get vineyard space all ready for projects, when we actually start running projects. I've also been working on a fruit quality project. For this project, I've been following grape clusters throughout their different developmental stages. Once they hit a stage called veraison, I bring them into the lab. This stage also is when the grapes become soft. Once the clusters are back in the lab, I remove the berries from the cluster and put them into our texture machine. This machine mimics a person's chewing, and the machine gives us 3 important values: springiness, chewiness, and gumminess. During the winter, I prepped for the following spring and summer. I prune back our vineyards as well as propagate dormant cuttings for future projects. I've also helped run a mealybug project here on site. For this project, we've looked at multiple cultivars of grapes and their resistance or tolerance to mealybugs. To quantify this, we actually count how many stage 1 and 2 crawlers there are, how many stage 3 crawlers there are, and how many adults. It's a very tedious project. A lot of counting, your eyes definitely get tired, but it is definitely a fun project. You kind of get a bit attached to the mealybugs, actually.
So I've pretty much always known I wanted a career in science. I've always been around agricultural sciences, and I've practically been gardening since birth. Even when I was a freshman in college, I knew I wanted to get a PhD. I just wasn't really sure what I wanted to get it in. I knew I wanted to be plant sciences, but I thought that I would have to choose between studying the genetics of plants and getting a degree in plant biology and getting a degree in horticulture, which is where you're growing plants. But neither of those was really a perfect fit. And I didn't know that things like plant pathology were even a degree. Fortunately, I found some great mentors, and I had the opportunity to do a few internships, work in a couple different labs, and eventually I found myself in a PhD program in plant breeding genetics and biotechnology, with an emphasis in plant pathology, which actually turned out to be perfect for me. I've always been interested in plants and how they respond to stress, so this was just kind of marrying all my interests together into one. These days, I think my story is a little atypical. I mean, I've pretty much always known what I wanted to do. It was just a matter of exactly how I was going to get there. And not everyone is like that, which is fine. But I do think what is important is why I stay in science. And yes, it's in part because I love science, but it's because I find it challenging. I'm always learning new things. I'm always finding new things to study. I also work with amazing people. I have some great collaborators who are fun to work with, and that's what makes part of this job so fun. And maybe most importantly, the work I do makes a difference. I feel like the work I do is making the world a better place. And you never know where the next project will be, the next thing you'll learn, or what's hiding in a grapevine. I'm not just a plant scientist. I'm a plant pathologist, an entomologist, a computer programmer, a writer, and so much more.
My name is Jeff DeLong, and I'm a biological science technician at the San Joaquin Valley Agricultural Science Center in Parlier, California. I have a master's degree in plant pathology, and I participate in research that involves population genetics of plant pathogenic fungi. So, I went into science because I was genuinely fascinated with the natural world around me. I'm a nature enthusiast and conservationist. And because of this, I wanted to know more and actually understand how the natural world around me interacted and functioned. While seeking my degree in biological sciences, I began working in a conservation genetics lab. It was here that this lab sparked my interest in the molecular approach to understanding the natural world around me. So I wasn't really sure where this career path would take me, and that's when I took a mycology class that sparked my interest in fungi. I quickly started volunteering at the USDA ARS lab in Athens, Georgia, studying a fungus known as Fusarium. Most of the work involved facilitating isolate crosses that created and screened for gene knockouts. I loved it. From there, I immediately ended up pursuing my master's degree in plant pathology. My research there looking at population structure of the gladiolus rust fungi. And from there, I never looked back. So, of all the different projects I've had the opportunity to work on in this lab particular, I would say Botrytis cinerea has dominated my research and time here. This project involves looking at population structures of Botrytis as it relates to the many cultivars of Vitis hosts. By understanding the genetic diversity that exists in these populations, often in respect to different fungicide applications, we're able to identify if resistance or fungicide resistance is occurring within these populations. This is a critical tool for strategizing and developing disease management strategies. In regards to the Botrytis research, a lot of the work involves DNA extractions from the fungal isolate tissue and PCR amplification. The process involves aliquoting a small amount of extracted DNA into a reaction well with a customized PCR master mix reagent. This reaction is then run in a thermal cycler under customized repeat temperature conditions to amplify specific sequences of the isolate genome. By analyzing the sequence differences, we are able to better understand the genetic differences that occur among and between the isolates. So, one of the biggest obstacles I had to overcome for myself was finances. As a non-traditional student, pursuing my degree required that I worked while pursuing it. For myself, this came with sacrifices such as missing time with friends and family. But to help me overcome these struggles, I had good emotional support from my family, and I was even inspired by many of those I worked with in the lab around me, making friends with both my mentor and coworkers. Often able to joke on, long nights in the lab and just getting research done.
What got me into science is when I was in high school. I did a 5-week-long science experiment that studies the effects of bacteriophages on bacteria. However, what really got me interested was that my coordinator at the time allowed me to use blood agar instead of regular soy agar, and it showed that bacteria can become pathogens by changing their environment. And it was there that really just boosted my interest in science. Both of my parents immigrated into the United States before I was born. However, since they were from El Salvador, they never received anything higher than a junior high education. This made it difficult for me as a first-generation student to really understand how college works. Luckily, I was in a college program when I was in high school, but it was still difficult as I wasn't able to grow up with a pair of parents teaching me how to read in English and write. However, I was able to overcome that obstacle, and I'm able to work in a lab as I am today. Something that I learned about working in an actual lab is that projects are a lot more longer term than expected. One of the projects that I work on started back in February. Another thing that I learned was that labs are very connected with each other. There were some labs from Georgia that were helping. There was Napa that would just send in their samples to get tested. So I thought that was very interesting and different than the regular in-class labs. The project I'm currently working on has to do with fungicide resistance on botrytis and alternaria due to the fungicide programs that they do on powdery mildew. It's a side project to another project that we have. I help prepare media using the 3 different fungicides that we work with, and also I sometimes help recover isolates and transfer botrytis from one plate to another. I also help evaluate the fungicide resistant test results, which is basically where I measure the diameter of the mycelium that are growing against the fungicide.
I'm a postdoc in Rachel's lab in the USDA ARS. I'm interested in plant pathology and mycology. My project focuses on evaluating the effects of powdery mildew fungicide spray programs on the non-target fungi for their fungicide resistance. I also study the microbial communities under different fungicide spray programs in the field. To do the fungicide resistance test, we transferred the mycelium pieces from the fungal isolates to different fungicide amended plates and control plates. Then, these plates were set in a box in the dark to let the isolate grow for 7 days until evaluation. The resistant or sensitive phenotype of an isolate to a certain fungicide is determined by measuring and comparing the diameters of the fungal colonies on fungicide amended plates and control plates. I'm always fascinated by the nature, and I like to understand why and how things work. For me, plant pathology is an area that I feel I can do research to help understand and solve problems that farmers growers are facing, and that's meaningful for me. Sometimes I have to learn new techniques and all the necessary knowledge and concepts behind them. If there is no one around familiar with it, I have to seek resources to solve the problems, like reading a lot of papers, networking with people, but it's a very good self-training experience.
The best advice that I could give someone going into science is be prepared to fail, with the caveat that failure isn't a bad thing. Science is a little bit about luck, it's a little bit about being at the right place at the right time, but it's also about taking chances and exploring. And if you're exploring and you're trying new things and you're trying to answer questions that haven't really been answered before, you're going to fail. I can't even begin to tell you the number of times I failed. whether it's an experiment that I planned poorly or somebody else messed something up. Sometimes it's just environmental things that you have no control over. All of these things can make you feel like a failure or make you feel like an imposter, like you don't belong, but that's normal. The important thing is to not let that failure stop you from moving forward. My approach isn't everyone's approach, and I'm not saying this is the right approach for you, but in general, I acknowledge my failures, but then I try to learn from them. I try to pick up the pieces, whether it is salvaging what I can from an experiment or starting from scratch, depending on how bad it is. But I'm always trying to look for ways forward, ways to fix it, ways to make it better the next time. Because if I sit there and I dwell on the fact that we just lost thousands of dollars because this didn't work, it can be debilitating. And so that's why, if you do plan on going into science, be prepared to fail, but know that it's okay. It's normal. And if you're not failing, you're probably not learning anything new, because we do learn a lot from our failures. I know it's cliche, but there is truth to it. So I won't regale you with all of my failures, because trust me, there are plenty. 2 of the more recent, just in case you think that maybe you don't make them as you get older, these are 2 that happened last year and the year before. We set up an experiment and we had thousands of plants set up to be able to do a study, and they all died. The greenhouse fan stopped working, and so the greenhouse went from being 80 degrees to 120 and killed most of the plants. And then there was this other time that I had a great idea of creating a mini greenhouse within the screenhouse, and we accidentally set it on fire and burned all of our seedlings. Yeah, that was a special day for us.
My name is Ben Halleck. I'm a graduate student at Fresno State, and I'm a lab assistant here at the USDA. One of the things that got me into science was just figuring out how things work and seeing the inner workings of things. My project is looking at Botrytis cinerea, and it causes gray mold in strawberries, and we are looking at how isolates from different hosts affect strawberry, and whether the isolates are how they respond to fungicides. We're taking isolates that have been previously identified as either fungicide resistant or fungicide sensitive, and we are applying them to strawberries and applying fungicides related to each isolate to see how they respond on strawberries. The reason I went to get my master's degree at Fresno State was better opportunities for career in research and development. I had a bachelor's degree in agriculture, and I was kind of limited to sales, and I thought that getting a graduate degree would be good for my career to get more into the research and development side.
So at this point, you've met a bunch of the different people who are currently working in my lab. You've heard their stories, you've heard about some projects that they work on, obstacles they've had to face, and hopefully that gives you a little bit of a better idea about the different types of people who work in a lab and their roles. And the one thing I want to leave you with is that the labs and the stuff that we talked about today, this is just one example of a lab. Our lab is a translational lab. But there are highly molecular labs where they do purely lab work, and they might work on cancer genes. You have labs where they don't even have a physical lab bench anymore. They're doing all their work on computers using DNA that is from sequencing centers. You might have applied labs where they almost never go inside the building except to bring in field samples, and most of their research is done in a field setting. So whatever your interest, there is a lab that will fit you and your research interest. And the other piece is that you need to find a mentor who also fits your style. So one example from my own life is when I started my PhD. So when I started, I was working in a different lab, and I was there for about 4 years. And after 4 years, I decided to quit and just get a master's instead. It wasn't that it was a bad lab, it just wasn't a good fit for me. I didn't feel like I was getting the mentoring that I needed or the direction. I was confused a lot of times, and I just didn't feel like my project was making progress, and I wasn't getting the support that I needed in the lab. So I ended up finding another lab, started a PhD there, and obviously at this point I graduated, I have a job, and my lab that I'm in right now is doing quite well. But that just goes to emphasize and to show you the fact that just because you're not doing great in one lab doesn't mean you won't do well in another lab. Not every lab is a good fit for every person. So with that in mind, as you move forward, I encourage you to look at the research that's being done in a lab, but also to look at the people, because they're really what's going to help make or break your success moving forward. So with that, thank you all for listening, and we'd be happy to answer any questions you might have.